All right, so what I do want to start with is I do want to start with making myself do what I ask you to do. I'm going to do a 10-minute summary of the paper. So I'm going to go from beginning to end of the paper, and I'm going to do it in 10 minutes. Then I'm going to go back and I'm going to talk about stuff in more detail. That'll be about how do you read a paper like this. So I will start with my attempt at a 5-slide version. I tried to get myself, I think even this I didn't quite follow my own rules. But I will show you, after that we'll go back and I will show you some sections from the paper to help you translate. I like to take notes on the paper itself, and as far as open note goes for the quizzes, that's actually allowed. Or you can keep it on a separate sheet of paper. However you want to do it, you just want to have some way to exactly point to things that are on the paper. Now many of the concepts are similar, so like this is different. And it's different in that it's older, and also that it's actually using a combinatorial strategy, which means that they are not really rationally designing. Mostly they are doing randomness, and they're just basically making a whole bunch of random mutations and seeing what works. But they do have some structural organization to that randomness, because if you look at how many amino acids could be in a sequence, you can't, even a short sequence, you can't fully exhaust how many amino acids are in it. You have to constrain it somehow. And so they actually do this in this paper. It's structurally specific, and they interpret it structurally. So I just want to say, this is actually combinatorial design more than rational design. It does have some rational elements, and that's the kind of thing that I want you to be involved with. Okay. All right, so you want to, let's take the time. It's 3:17 right now. We'll see if I can get this done in 10 minutes. So this paper is about, it's actually a fairly old paper, 2000, and it gave an early example. It's fundamental in how protein design came about. It's one of the first very successful examples of protein design, and they weren't just designing something for the fun of it, they were designing something to answer a particular question. In fact, their question came from the fact that you see convergent binding. You basically see the same binding site on one side of the interaction. And then on the other side of the interaction, you see 4, 5, 6 different things that pair off with and interact with that exact same binding site on the bottom. The binding site on the bottom is the Fc binding site. It's part of antibodies. And what they did is they asked a design question to say, is this site really unique? Was this picked for structural biochemical reasons? Or was it just that these all just happened to converge on this spot? They found out that the convergence is biochemical in nature, and there is a chemical reason why they chose that site over and over again. What the convergence is chemical. And so the thing about the site that I was telling you about, the Fc region of the antibody is on the bottom of this, shown in blue, and on the top, shown in red, are 4 different proteins that bind to it. And you see that we have, if you compare the red proteins on the top you have very different structures. And yet you see where they're interacting with the blue on the bottom, they're all interacting with the joint of the 2 domains. And so you can see that they're all binding the same spot, and these are 4 natural examples derived from nature of different proteins, different species, different systems. And yet they all bind the same spot. You can see that they have very different structures from this. So what they wanted to ask is what would happen if maybe they're keying in on the same chemistry, okay? Maybe they're all asking, they're all a charged interface. And you look at protein G's binding interface, and it's actually a charged polar interface. Okay, maybe they're all like that, but no, they aren't. If you look at protein A, it's also it's a hydrophobic interface, hardly a charged or even polar residue to be found. And so somehow protein A and protein G are not just latching on to the same chemical reactions. There's something that is more complex than that. And that's one of the reasons why we were like, well, is there anything that's biochemical in nature? So the way that they asked this question is they made a 5th binding protein to this site, and they basically set up a system where you evolve a peptide, and you say to the peptide, the only thing I want you to do, you can be anything you want to be, You just have to bind this blue protein that we have. And so the way they did that is with phage display. This is really helpful because it connects the bacteriophage has a DNA inside that is with a protein shell on the outside. And you can program the DNA on the inside to express different protein shells on the outside. But also you can select for the protein shells that bind, wash off the ones that don't bind, and then sequence or amplify the ones that do bind. And then you can randomize them, and you can do it again. And you can get a little better each time you do this. You can optimize the binding. Basically, this protein library is randomized. And you can randomize it, bind it, select the ones that elute the ones that actually bind the column, and then randomize it again. And in this way, you can have several rounds of selection. It really is like evolution in the bottle. So bacteriophage will infect bacteria. They're incredibly cool. They're easy to modify, and they could randomize them. They didn't do it completely randomly, though. They joined the 20 amino acids in a loop with a cysteine at either end. Once they did that, they did several different rounds, and they came up with these two different peptides from different rounds of selection. And so you end up with, they call this one FC1 and FC2, and notice that they have certain similarities to them. Of course they have the cysteines because they made them have the cysteines, and they randomized the other residues. But when you look at this side, the right side of the sequences, you can see that despite all the randomness, they both develop this GELVW sequence. And so both of these elements were preserved. Also, the cysteine spacing was allowed to vary a little bit. So the fact that the same cysteine spacing was developed by both rounds of selection let them say, okay, we need to keep this. We need to keep the residues that are highlighted. And then we'll randomize everything else and do a third round of selection to see if we can get it even better. And it turns out that they did get it even better. They bound really tightly as the much bigger proteins with bigger interfaces. So that means that it was a success. They crystallized it, and they found that this little red peptide bound in the exact same space. You see the blue parts right here. It's binding to the hinge between the two domains. It converges to the same part of the FC domain. And so success. So now you want to zoom in, and you want to look at it more closely. There is something about this site. Remember that we gave it the whole protein to bind to, and we made it random so that it would bind anywhere. It could have bound anywhere, and yet it bound to the same place as all the other ones. There is something biochemically special about this area of the blue protein that things like to bind to it. And so when you zoom in on that, you see here's the crystal structure of the peptide itself. You can see the cysteines bound in a loop. You can even see the tryptophan that's in the GELW sequence that they had in the middle there. And they showed that the binding sort of converged to the same part. Now it's important to point out that you can't conclusively identify hydrogen bonding interactions. The resolution was just too low. But they went ahead and identified them anyway. So it appears that 8 hydrogen bonds are formed, including those involved in salt bridges. So there are salt bridges involved, there's hydrogen bonds, and there's some hydrophobic interactions involved. It looks like every type of interaction is involved in this phage display evolved peptide. Okay, so it's important to remember we're not quite sure about where all these residues are. We're not quite sure that there's 8 hydrogen bonds, but this is definitely sufficient to recognize what type of binding it is, what the general structure is, and that it's a mixture of all the types of interactions. It's forming all types of interactions. And so if you look at the groove that basically binds everything, it binds all 5 of these proteins, you can see that there is sort of a little trench where all the proteins like to bind, and then there's sort of the edges of the trench where you have you have some bulky residues that are providing the edges of the binding site. So it looks like it's a little notch where where proteins like to bind to. And you also have some interesting options for mild interactions. you don't have like a really big lysine, or if you look down here, you have an arginine and a glutamine, but those are on the edge. In the middle, you have a histidine asparagine, and then you have these bulky sort of hydrophobic residues, not completely hydrophobic and not completely the largest hydrophobic ones. But the other thing that you see on top of those is you have methionines at the middle. That's unusual to have two methionines in the very center of the binding site. So I propose that this binding site works from a combination of things, having the methionines, the residues pointing up, and some of them are hydrophobic, some of them are polar and combined things. Nothing too extreme. It's just a combination of good medium interactions rather than leaning everything on one kind of residue. If you zoom over, you can see how this site compares to the other sites, and you can see that the other sites are making contacts in this region, but they're doing it in totally different ways. So it looks like this combination of amino acids allows this site to make versatile interactions with different other proteins. So you can see that none of the other yellows line up. But, and also the site, the groove is always maintained. But the edges of the site, you see the sides of the site, they can be twisted differently. If you look at 2 here and 2 there, that's different. 4 here is up here and 4 is down there. So you have, what happened with this site is it looks like the edges can move, and so you have limited rotation. It's not a huge conformational thing, and that's one of the things that you noted about the word conformational when we started. So the other thing they wanted to do is they wanted to analyze the site and say, can we put a number on how special this site is? Why so many things want to do it? And they noticed that it was pretty non-polar and very accessible. Like it was out on the surface of the protein, and yet it was non-polar. There's a lot of hydrophobic residues, especially relative to what you would expect on the surface of the protein. And they actually came up with a ranking where they ranked all the patches on the protein. This protein comes out in definitely the upper standard deviation for accessibility, but lower standard deviation for polarity. So it is both highly accessible and lowly polar, if that's a proper word to use. Which suggests that this is a unique site because it manages to be both accessible and hydrophobic at the same time. It's definitely both of those things. It's not the absolute most extreme, but it's probably extreme in how it combines both of those contradictory things at once. So in that sense, it's walking a fine line between accessibility on the one hand and hydrophobicity on the other. And the site manages to walk a fine line just like you're walking along the ridge of this, where you don't want to be too inaccessible, because then you won't be able to reach the binding site. You don't want to be too hydrophobic or too hydrophilic, actually. Either one, and you would have dominating one kind of interaction that would cause your binding possibilities to be limited. And apparently, this does not want to be limited. So, my time's 11 minutes, so I would go a little bit over, and by the way, I'll have a little time card so you'll be able to tell when your time is almost up. But that would be an example of a 10-minute talk on that. There's so much to say, right? You're going to have to leave something out. But that's what I chose to stay. And now I want to do all those things.